Thank you everyone for joining. Welcome to our webinar on biofertilizers, brewing your own biological workforce. We'll jump straight into it so we have heaps of time for questions. My name's Courtney. I'm the Peer Learning Manager at Soils for Life. I'll just try and change slides. Just a little bit about Soils for Life. We support Australian farmers to regenerate soils for resilient businesses, landscapes and communities. We have an amazing library of regenerative case studies, which I encourage you all to check out. And we also run lots of events like these. This project is part of our cropping resilience project, which is all about supporting Australian grain growers specifically to build soil health and drought resilience. So yeah, again, we're running lots of events, lots of webinars, and we've just released 2 practice guides on regenerative cropping practices, one being on biological seed treatments and the other on multi-species cropping. We also run some cropping discussion groups, which I can share links to in our follow-up email. Oops. Before we kick off, I just want to acknowledge that I am standing on Bungarang country today. I'm here in Rutherglen, Victoria, and I'd like to acknowledge the traditional owners here and from all of the lands that you're all zooming in from. In terms of housekeeping, ask questions in the chat as they arise. Please stay on mute if you can. We will have a Q&A from 6:30 p.m, so you're welcome to jump off mute and we can have a bit more of a discussion then. We're recording this webinar, so please keep that in mind, and we will be trying to keep to time. Marco and Callum, our guest speakers, will only be speaking for about 30 minutes each. So we appreciate you helping us with that. And yeah, if you want to find out more about the Cropping Resilience Project and what we're doing at Soils for Life, please jump onto our website and you can find out more. So I'll hand it over to Marco now to kick us off. Marco is a biological agronomist and a business development manager at Nutri-Tech Solutions on the Sunshine Coast. We just ran a field day with Marco and a local farmer, Will McInnes, the other day. Which we had some fantastic feedback on. So we're delighted to have Marco join us again to share his kind of insights working with Nutri-Tech and as an agronomist. All right, I will stop share. And now you should be able to. Thank you, Marco.
All right. Okay, thanks, Courtney. And also thank you, Soils for Life, for inviting me to talk about a very important strategy on biological agriculture to regenerate our soils. So I'll get straight into it because there is a lot to discuss. Biology is a very complex field, so the more we go through aspects and how to better harness our biology, the better. So quickly running through the agenda. I want to discuss about what the terminology of biofertilizers, why we do it. I put it benefits versus benefits because yeah, it's a very good strategy. Also the soil and plant improvements that we can expect from it. What are the different type of biofertilizer methods that we can do? We can do aerobic, anaerobic, and extraction. Also, a very important one is to talk about major critical aspects when we're doing our fertilizers, what we have to control in regards to temperature and so forth, oxygen to efficiently do our biofertilizer. DIY biofertilizer, I'll probably leave more to Callum, but I'll probably just put some steps on how to do it yourself, how we can monitor our biofertilizers. I think sometimes it can be certainly kind of unknown if we're doing the right thing. So just some tips on the process to see if we're doing the right thing. application methods and rates, considerations, and also how we can monitor if our biology or biofertilizer is doing a good thing for our soils. So the first thing is, what's a biofertilizer? On the name we got bio and fertilizer, which means we got microbes, and we can also have fertilizer. You can have one of the other, always having the biology as well. In some situations, it only contains living microorganisms that they can actually promote a better bioavailability. So if we apply this biology to our seeds and as a seed treatment, liquid inject, foliar spray, or fertigation, including anywhere in the range of microbes from bacteria, fungi, protozoa, and algae, they can increase that bioavailability. It can be in different ways, improving the soil organic matter decomposition. It could be also by producing acidic compounds that release locked up nutrients, and also using nitrogen from the atmosphere, making it available to our plants. Also, one thing is, I think some of you are familiar with Elaine Ingham. She does a big statement saying that a good compostee can control any type of disease. It's a big statement, but if we do the right biology combined with really good strategies on our farming strategies, this statement can be totally true. So why do we do it? The first thing is why we brew microbes, why we ferment microbes rather than just doing the direct inoculation of microbes. The first thing is on our pockets, to reduce the application rates. The idea of brewing microbes, fermenting microbes, and doing a fertilizer is to increase the batch further. So when we brew them, we can increase by 10 times. And when we increase that by 10 times, we can reduce the cost to, depending on what you're doing, if it's a seed treatment or fertigation, application rates can be anywhere from 20 cents to $15 per hectare, depending on inoculant and what you're doing, of course. The beauty of it as well is the versatility. So when we brew microbes, we can select specific species that they have specific tasks to something that we want to target in our soils, or we can focus on something a little bit more multi-species, something more broad spectrum, where we have a combination of different types of microbes that can play different roles in our soil. Also, as I mentioned, we have a lot of different ways of doing our biofertilizer. We can do aerobic brewing, we can do anaerobic fermentation, and even microbe extraction. And still on versatility, there are many different ways we can apply that. We can do seed treatment, fertigation, and you name it. The other benefit as well is to increase the inoculation efficiency. What we can do with the microbrewing is stimulate our microbes to go in what we call the endospore form, which is a survival structure of the microbes. So they can resist to any extreme weather conditions such as heat and even chemicals. And by doing so, we can increase the efficiency of the biology itself. Looking to some of what to expect when we put out our biology. What are the soil improvement and plant improvements we would expect? The first thing is, by increasing biomass and biodiversity, we're going to have a much healthier soil that can be disease suppressive. And how does disease suppression work can work in different ways. One is creating a biofilm. It can be either on the leaf surface or on the root system. So when you inoculate biology, you improve that biology in the root system, it creates a shield around the root system, and then potential pathogens, they can't penetrate and cause any damage to our plants. Another interesting one is induced systemic resistance. So if you have a look at this picture on top here, when we have a biology and creates a synergy relationship with the plants, it starts producing some compounds that stimulate the plants to produce some protective components, such as jamonic acid or salicylic acid. This picture here in the bottom is a very interesting trial that we've done in an apple orchard, and we treated the left one with EM. So EM For those ones are not familiar, it stands for effective microorganisms. So it's a type of fermentation where you have different group of bacteria such as bacillus, lactobacillus, streptococcus, streptomyces. And we applied on one plant, treated them in the soil application, and in the other one we didn't, and powdered the one that wasn't untreated, powdered immediately took off, whereas the other one had the protection by the induced systemic resistance. So this is a big one coming from a big benefit from biology. That's where the name comes from, biofertilizer. So it's increasing nutrient bioavailability. And we can do that in a lot of different ways. So the first one is introducing microbes that they can help increasing the soil organic matter decomposition, including lactobacillus and actinomycetes on a combination of the EMs that I just mentioned. I'm probably not gonna get really into EMs, but if you wanna get more questions later on, I'm happy to discuss about EM fermentation. But if you introduce these microbes, they can break down organic matter and make nutrients available. This is on the top right corner here, is an experience with a farmer. He used macadamia husks and skins to make a compost using those EMs. And within 6 months, you can have a really good breakdown. So if you apply these microbes in your soil, in your crop, you can have a similar result. Nitrogen fixation, using nitrogen, free nitrogen from the atmosphere is a big win, using Rhizobium species and Azotobacter, for example. We also have Bacillus subtilis species, which is a very important one to increasing phosphorus availability, for example. So they can exude. If you have a look at this picture on the right bottom here, it shows how it works. So the Bacillus species releases some organic acids that goes into the clay colloid, which is the brown particle here, releases this phosphorus and make it available to the roots. The roots don't have this potential to create these acidic compounds to make this phosphorus available. So in soils, red soils with really high iron levels where phosphorus is locked up, Bacillus species is one of the big friends to improve the phosphorus availability. There is a concept called MEND, which means microbial enhanced nutrient delivery. This is a concept that works in the roots, but also on the leaf as well. And for example, on foliar sprays, one thing that I always recommend is the microbes behind the minerals. So the microbes, because when you apply with the microbes in a foliar spray and they colonize on the leaf surface, they start reproducing themselves and growing, expanding, they produce carbon dioxide. And the carbon dioxide elicitates the stomata, which is the structure of the plant where the nutrients go into the plants. So when you use microbes, you can increase the uptake of the nutrients that you apply as a foliar spray, for example. Another example of nutrient bioavailability increased by microbe inoculation is mycorrhizal fungi. This picture down at the bottom here shows exactly what happens when you have mycorrhizal fungi and when you don't have. So the mycorrhizae can expand the area that the roots, they can scavenge nutrients in the soil. So that's a big player for you. Improve soil structure, so increases the soil organic matter decomposition. Biology also produces glomalin, which works as a glue effect, keeping soil aggregates together. In a soil where you have good biology as well, especially bacteria and protozoa, for example, we have a lot of earthworms, and earthworms are a major player for soil structure because they create the channels to improve drainage and aeration, and all this combined allows us to have increased root development. Interesting usage for microbes as well is for soil detoxification and bioremediation as well. So microbes, they can use chemicals as a food source. So carbon, nutrient, and sulfur from the chemicals can be used by them to produce energy, and they can use enzymatic breakdown. So they produce enzymes also that they can break any chemicals that could be residue. So for example, Pseudomonas is a very well-known bacteria used for soils where they have been plugged with atrazine. So Pseudomonas uses the carbon source from atrazine degrades it, and the residue impact from atrazine is gone due to the breakdown from the bacteria. Stubble digestion, this is probably one of the key strategies for a long term to build up this organic matter in the long term. And when we apply biology, we have elements such as bacteria and fungi. They are very important for that strategy. If we try to keep it simple, the bacteria would be considered sort of the initial decompositors of this thermal digestion, and then the fungi would be the one that comes after and the main player to break down that stubble and accumulate that stable carbon in your soil. So in broadacre situation, in grazing systems, a usual fungi to bacteria ratio that we aim is a 1-to-1. That's usually for annual crops. Elements that are added for that would be trichoderma, bacillus species, aspergillus, and streptomyces, for example. This here down the bottom is a result that I'm going to talk more about the microbiometer later. But these are two different results from two blocks. They had the same crop cycle for the previous 5 years. But they had slowly different strategies, and he was using microbes through liquid inject in one. So the first one, poor stubble digestion, very low readings of biomass, and you can see 165 is very low, and the fungi to bacteria ratio very far from 1-to-1. So the stubble was still standing even after 7 months. And the bottom one, he was using more microbes. He had kaolin, and biology was 3 times greater, and the fungi to bacteria ratio was 4 times greater as well. So the stubble was all not broken down, but it was all in contact with the soil, and you can see microbial activity degrading it. All right, different methods of biofertilizer. This really going to depend what you want, what you're chasing, and what sort of structure you've got in your farm. But there are a lot of different ways of doing it. First one is aerobic brewing. So we use air in an open system to brew strictly aerobic microbes. We can target species-specific microbes, but also we can have a more broad spectrum type of brew as well. The good thing about it is very quick. It takes 1 to 3 days, depending on what you're brewing. Bacteria usually takes 20 hours, but if you're brewing protozoa, for example, it might take up to 72 hours. Fermented biofertilizer, I think that's Callum's going to talk more about his today. So this is strictly anaerobic microbes, or even facultative. So facultative means microbes that they can adapt to aerobic and anaerobic environments. So the EMs that I was talking just then, they are part of the fermented biofertilizers. There are types of biofertilizers, anaerobic ones, that you can take 5 days, but it can go up to 60 days, depending on what you're doing. There is also the microbe extraction, where we just basically use air to extract the microbes that are part of the compost or a vermicast. The only downside of it is that you're not replicating the microbes, so you're not increasing your biomass, but you are actually getting a much higher biodiversity when you do this kind of methodology. Okay, this is a very important topic to discuss major aspects. So the finished brew can vary depending on a lot of brewing aspects. And for us to really be consistent and have an effective biofertilizer, we have to understand the dynamic of biology, even aerobic microbes, anaerobic microbes, and provide them the right environment. So it's all about giving them the right environment. If you do that, your biology is going to be able to breed and reproduce to greater numbers. First one is the temperature. So ideal temperature for bacteria, for example, they prefer warmer temperatures around 25 to 35. Fungi a little bit cooler, between the 15 to 25, and protozoa in between the both. That doesn't mean that they necessarily need these temperatures, but it's always the best to target these temperatures. If the temperature is not ideal, the brewing time might be extended. So bacteria, instead of 20 hours, might take 40 hours. And that happens because metabolic rates, a rule of thumb that we use is that metabolic rates of microbes, they double for every 10 degrees. But it doesn't mean that if you go up to 40, 50, 60, 70 degrees, you're going to be speeding up the process and having more microbial reproduction. But actually, it can compromise because you can get to temperatures which degrades the proteins from bacteria. So sticking to that temperature range is fundamental. Food source, second point, the same way that they prefer different temperatures, they also prefer different food source. Bacteria, they're much simpler microorganisms, so they prefer simpler sugars, like amino acids, molasses, fish emulsion, fulvic acid, and even refined sugar. Whereas fungi, they're a little bit more complex, so they prefer food source that they have a larger chemical chain, such as kelp, fish hydrolysate, humic acid, and the others listed. I think Courtney's going to make these presentations available later, so I don't have to go through all of them now. Another rule that we have to follow in regards to food source is never exceed 1% of the brew volume. So if you're doing 1,000 liters, usually stick to 10 kg or 10 liters of the food source. If we put too much food, we can actually induce a really quick spike on the population of the microbes, which can, for example, in an aerobic environment, it can deprive oxygen, and then can be a big problem for the population because there is no oxygen available for them to keep going. Inoculation, if you buy a commercial product, always pay attention to the CFUs, which is the concentration, the biomass of microbes you have in the product, specification, biodiversity. So this is what's going to lead your whole brew, the biology that's going to dictate what you're doing as a biofertilizer. So this is extremely important. Oxygen depends what you're doing. If you're doing aerobic brewing, ideally we should have what we call dissolved oxygen levels, which is basically the concentration of oxygen around the 8 parts per million, and minimum 6. So when you have a lot of population of fungi or bacteria growing in your brew, this availability might tend to lower a little bit, but then it stabilizes and goes back to close to an 8, between 6 and 8. If it goes for below 6 for too long, you might end up having contamination on your brew. And of course, anaerobic brewing, no oxygen. So this airlock, this picture we've got here is just an example of an airlock that it's used on anaerobic brewing to allow gases to get out of the tank, but no oxygen can get in because we're focusing on the anaerobic microbes. Okay, water is a very important point as well. So they have preferences on pH. Bacteria prefer more alkaline environment, and fungi, they prefer more acidic. So we can adjust our water when we bring microbes depending on what we're targeting. Electrical conductivity is a term that shows how much salinity we've got in the water. So usually target below 700 microsiemens per centimeter. That's usually a target for not impacting the biology. And of course, no contamination. Hardware is usually an underrated topic, but I think it's a very important one. And probably the most important message is try to get an equipment, if you're doing it yourself, easy to clean, because hygiene is crucial. And if it's too hard to do it, you won't do it. I found that tanks like this one on the right, I think probably Callum is going to be able to show what he's been doing as well. But the conical shape of fermenting and doing the biofertilizer, it's usually easier to clean. The IBC, sometimes they can be a little bit of a struggle to clean on the corners. And also, so this is on the hygiene side of things. And then on oxygen availability, it's if you're doing an aerobic brewing, just make sure that the system you have, this is an example of a farmer that did himself. He used some Venturi outlets here to produce enough oxygen for the microbes. And then using correct pumps, usually we try to use a diaphragm pump rather than a centrifugal pump on aerobic brewing, because physical biology can be sensitive to physical impact. And make sure on the equipment, if you're doing an aerobic brewing, you have a proper airlock. There is no air getting in. Quickly go through it. The first thing when you're doing biofertilizer, I just mentioned, first thing is sterilization. There are a lot of different ways to do it, and probably the most simple way is using chloride bleach, using 50 ml per 100 liters of water. There are other alternatives such as quaternary ammonium, which is an interesting one because it's only effective on microbes while you have moisture. So if you use quaternary ammonium, for example, and you clean your whole tank, your equipment, and you let it dry, you're making sure that all the active ingredient is gone and it's not going to impact your brew. Then we add the water, and always checking to those aspects that we mentioned before: pH, electrical conductivity, temperature, no contamination. And then add the food source, add the inoculant, and then we check if the dissolved oxygen levels they're actually ideal. After all the inoculant and food source has been added, it depends on the time. So bacteria, as I mentioned, simpler microbes, they take 16 to 24 hours in ideal conditions. Fungi, a little bit longer. Protozoa and fermentation, any time from 5 days onwards. So how can we monitor to see if we're doing the right thing? During the process, one thing that we can do is easily use a pH meter to see if the pH is ideal for the bacteria brewing or for the fungi that we do in the biofertilizer. There are also equipments that you can use, which includes a dissolved oxygen meter to measure if your oxygen is fine, and then the airlock system for anaerobic. Then, to see if we did a good biofertilizer, a few things that we can do is, so for example, effective microorganisms. To know if you got to a point where it's finished, all the biofertilizer has to drop to a 3.7 or 3.5 pH. So that's going to be the timing where you know that your EM has been brewed efficiently. Microbiometer is a tool that really helps to understand if you're doing the right thing. I'm probably going to open for you guys to have more questions later. I won't touch much, but it's a really quick tool. It takes only 30 minutes to measure. It gives you the biomass of biology and also the fungi to bacteria ratio. Another thing to look at to see if you've done the right thing is if you're brewing bacteria, for example, you would see some foam being created on top. And if you're doing a fermented type of brew, like effective microorganisms, you would see something like what we got on the right side here. It's a biofilm of yeasts that can be produced on top on the surface of your brew. Application rates. This is based on effective microorganism using lactobacillus species. The cost is going to vary, of course, and it doesn't include equipment. But if you're using a simple system using easy ingredients, you can end up getting a good brew for 32 cents per liter. And I'll leave it there, but the price can vary anywhere from, so if you're doing a seed treatment, 20 cents per hectare. So the price we got there roughly is for a ton of seeds. So if depending on the seeding rate, this price is going to change. But it really helps to cut the cost. And the idea is to put the biology where we really want it. And seed treatment, for example, is a really cost-effective way of doing it. Considerations when we apply. One thing is biology. Also, the same way that we did to create environment to brew them, we need to provide them an environment when we're putting out. So moisture is very important, trying to apply when you've got some good moisture in the soil, and also targeting bigger droplet sizes. It's important, avoid really hot days where it's very sunny. Some species like Metahebrisium and Bovaria, which are entomopathogenic fungi, they feed on insects. They're very sensitive to UV light, so trying to avoid these hot days, it's very beneficial. Filters, it really depends what you're brewing, but also you have to have a filter which this is going to be lined up with the type of nozzles you use and your equipment. So there is a big range over there. But at least 400 microns, because when you brew in microbes, you have to let them out, go out as well. If you go with really low micron sizes, some of the microbes might get stuck. Pressure is very important as well. Microbes are sensitive. Fungi are a little bit more sensitive than bacteria, so we usually aim to a 60 psi max. And bacteria, they can tolerate a little bit more, especially if you brew them into the endospore form. And another one is always send your microbes to work with lunchbox. So we always add a food source, like another fulvic acid, kelp, molasses, a lot of different food sources, depending on what you're brewing, the bacteria or fungi. Monitoring the outcomes, different ways. Again, you can use the microbiometer to tell if you're doing the right thing, if increase the microbiodiversity, fungi to bacteria ratio. Another way to assess, for example, is you doing a plant bricks testing. So if you apply the biology, they start increasing bioavailability of your nutrients, it should provide a response on the bricks levels, which is the sugar concentration on the plants. And the other one is assessing your stubble digestion. Just a quick example. This is a farmer planting cotton and over a barley area. And when we had a look at the bottom after he doing seed treatment and liquid inject, the stubble, that obviously wasn't through just one single year, it was a couple of years doing the right strategy, but we could see a really good stubble digestion. And just to finalize, we spoke about biofertilizer and using biofertilizers to improve our biology. The biology is one of the three elements of the soil that we really have to target to improve our soil health. So targeting the biology and putting it out, it's already a good step towards a really healthy soil. All right, thank you very much. I'll just hand it over to Courtney now.
Thank you, Marco. That was fantastic.
Just stop sharing my screen.
I think my main takeaway from that is sending your microbes to work with the lunchbox. That's very cute. Thank you again. That was awesome. Feel free to throw questions in the chat as they come up, everyone. We'll hand over to Callum now. Callum is a farmer and Vic No Till board member based down near Avenel in Victoria. And yeah, we're delighted to have you here, Callum. We know that you've been doing some awesome things with biofertilizers. So thank you for joining us.
Thank you, Courtney. Thanks for Soils for Life for putting this on and asking me to speak. Very honoured. That's very good. Marco, what a presentation. Hard one to follow. Lots of awesome information in there. You squeeze a lot in half an hour. So my name's Callum Lawson. I'm from Avenel, Victoria. There we go. We run about 1,200 hectares down here. We breed Angus cows. We rotationally graze them on time-controlled manner. Try and do a bit of cell grazing where we can. We're sown or average around 200 hectares of multi-species crops a year. Most of those are fertilised with bio-ferts made on farm. And there's only a few fertilizers we are buying in that aren't made on farm now. There's a little bit of starter fert and a little bit of urea, but I'd say probably 80% of our fert now is brewed here. To determine what we're actually brewing, we've started doing a lot more sap tests. Really interesting to do a total soil test and a normal soil test and then a sap test, and might as well throw the normal soil test in the bin. Seems to not really be very helpful. The information we're getting off the saps is actually what the plant isn't getting out of the soil. And yeah, you can have one extreme on your standard soil test saying you've got the 0, and then it's overdone in the sap test. So I'd recommend everyone sort of do a total soil and a sap to actually to look at that. And you always sap test with a shovel so you can see what your roots are doing as well. Most of our recipes that we use came from David Hardwick at Soil and Food. And then through Vic No Till, there's a lot of guys in that organisation who have been fantastic and don't mind sharing the odd recipe. A bit of an overview of what I'll be talking about today, what type of ferts what we make here, the reason why we use them, basic ingredients, a bit of a process of making some basic recipes, and the application. Types of bioferts or ferments we use and make here. The most simple one and cheapest one, which Marco talked a little bit about, was the lactobacillus. So it's based on rice, soaking rice for a week and getting the lacto off the rice hull and then brewing it with molasses and milk. Very, very cheap and easy one to make. Anyone could make that. Just it's large or as small as you like. We're putting that out with pretty well everything we put out as a foliar. The cab is the next one, which we base our actual bio-fertiliser on when we're adding our minerals. We also add a bit of manure and paunch to that when we can get it. Multi-mineral bio-fert with a lacto, which is our main fertiliser we make now, and we've been starting to ferment some fish hydrolysate. So the hydrolysate we've been buying, it's ground up fish, and it hasn't fully broken down. So we've been able to nearly get 150% extra on the fish that we're buying by brewing it, and it bubbles away very, very quickly. And we turned 1,000 litres into 2,500 litres. So the cost is pretty well half. Compost extracts. So we're using everything goes out with a compost extract here. Pretty much makes up the rest of whatever water we're missing in the tank, whether it be from a Nutri-Soil vermicast or a homemade vermicast or a Wild good compost. That everything goes out with that. This year we've been trialling a bit of chelating of our minerals because we hadn't brewed enough minerals over summer, so we've been chelating our own and had some really good success with that, lifting our sap levels of what we're deficient in. And the beauty of being able to test your plant on Monday, put your fertilizer on Wednesday and Tuesday, and then have your test back of within 24 hours is just incredible. So you could get 2 tests away in a week if you wanted to, sending it down to Tassie where we are. Anyway, the post system's been absolutely awesome and really powerful tool for us. Why do we use bioferts? Well, it was pretty logical to me that putting out these high analysis stuff wasn't really getting us far, and we're only putting 1 or 2, maybe 3 nutrients out. So we've sort of went down this path to make it a lot more efficient by attaching an actual mineral to a carbon so the plant can take them up. As Marco had a great slide on some of the costs, massive cost saving, like $5 to $10 a hectare sort of is we're what we're running on. Reducing that need for the high analysis fert, which is burning up a lot of carbon and not good for your microbes in the soil. It's an awesome job to do when it is a hot day in summer. We don't get many rainy days in summer here, so it's good to get out of the sun. And while the temperatures are up, as Marco also said, that temperature being 15 degrees at night, I think is crucial for brewing all this sort of stuff. So yeah, great summer job. We're brewing to our exact mineral deficiencies now, so we can pretty well custom make a biofert for any paddock we test, or that area of the farm that we know that's going to be deficient in a certain mineral. We can boost those levels up. So we're really getting exactly what we need, not just a bit of everything because we think it needs it. Increased plant and soil health with some of our bricks levels. We've gone from like a 9 to a 17 or 18 with some of these bioferts and biostimulants we're putting out. So yeah, insect damage and that is just disappearing pretty well. Some equipment. You can, don't get caught up on, you don't need big and fancy everything. You can start in a 20-litre bucket. You could do it in a milk container. Just if you want to have a go, just get out there and get your bucket and make sure it's airtight. But we sort of do it in IBCs or some tanks, but majority IBCs will be investing in some cone bottom tanks, I'd imagine, over this summer. IBCs can be quite challenging to clean, as Marco stated before. Just trying to get it out of the corners and it actually draining it all out can take just as long as brewing it, washing it. One-way airlock, super important when you're doing anaerobic bioferts. We do a pretty simple one of, I'll have a picture of it later on, but just a tube into a bottle of water. Clean water, so check your pH of your water, make sure it's not too hard, not too acid. We try and use rainwater wherever we can. Stirring and mixing equipment makes your life so much easier. These guys, the picture down the bottom there is an Agimate. They're made here in Australia and super simple, pretty cheap. You can get them that go on a drill, or you can get them with the motor like that. And awesome bit of kit. Filtering, you do have to filter it before it goes in the boom, no matter what, but you have to filter it a lot more if you're using paunch or manure. So we've got a vibrating sieve, which is probably a bit excessive if you're just doing kefir brew. Awesome for making sure you don't have any big particles that are going to block up your filters and your nozzles. And then if you're doing those sort of paunch or manure brews, you do need a trash pump because you block every other pump up, and it's not very fun when you've got a couple of 10-meter pipes full of solid manure. It takes a bit of cleaning. A few basic steps of some bioferts. Step one, making the base culture. So base culture can be used in pretty well everything. As Marco stated before, like your compost inoculants, it's pretty similar to an EM. This stuff, it's anaerobic, so you can add it to your compost, your hydrolysates, your bioferts. Yes, there's lots of things you can do. You can put it straight out over the top of your pasture as a fungal sort of disease remedy. Kefir is what is used, so we just buy it literally pretty well exactly that brand or whatever we can get at the time from the supermarket. Just go and buy a few boxes of it. You probably have to order it in because not many supermarkets actually have enough of it. It's mainly lactobacillus, but the advantage of kefir, it has a hell of a lot of diverse microbes in it as well. So I always think more the merrier, especially with all this stuff we're doing with biology. So that's why I do like to put that in, and as well as a bit of paunch or manure as well, just try and get a bit more diversity in. This can be used as, as I stated, cool. So a bit of a recipe here. So you'll get the slides sent out to you. It's pretty simple. Clean water, milk. We use milk powder. Heaps easier. I can have a pallet of milk powder sitting in the shed. I don't have to worry about it going off. As long as mice don't get into it, you're pretty safe. This is just for a 200-litre brew. The next slide's got the 1,000-litre brew on it. You can, if you've got a dairy nearby, you might be able to get some, milk off them, because you seem to go through a lot of it if you're making a few thousand litres. The next thing for us with a kefir is I've been given somewhere you can actually buy the mother for the kefir, so we'll be able to just buy, like, make the milk and add the kefir to it. You have to keep it and make it like an actual yogurt. So I think that'll probably be a fair bit of money saving as well. Molasses. Some people say you've got to pasteurize it. I don't think we haven't bothered, and we haven't had any issues at all with it growing a yeast or anything. Like, it's all pretty good. You can mix all these together too. So if you wanted to do half lab and half cab, you could. As I said, the paunch and cow manure is good if you haven't got any cows or sheep near you. Kefir you can just buy from the supermarket, which is pretty simple and easy. So there's the actual recipe. So that's just a simple one from Soil and Food. David's great. I spent a fair bit of time with him over the years doing a lot of different courses. So I recommend, if you're wanting to learn a bit of basic stuff, just go and see David up at Albury. So this is the next step. Cleaning. Then we'll sort of go through the actual process. This is one of the stainless vats we use to mix everything in. So we've got 3 of those now. So you can have multiple things going on at once. We mainly use that one now for compost extracts, but often when we're making a cab or something, we'll put it in there because it's got a big paddle mixer in it. You can stir straight molasses, no worries, without blowing a fuse or anything. Cleaning equipment, super important. The chlorine I haven't tried, but I like that idea. We use hydrogen peroxide and just sort of spray that around and let it all dry out and off gas, and then fill it up with water and off we go. It's probably really, it is challenging plastic, so it seems to sort of stick to it, a lot of this stuff. It is really, it grows on the side of it. You do need to scrape it sometimes. So as I said before, it is challenging with the IBCs, but at the same time, they're pretty cheap when they're getting everything delivered in it already. So it's whatever convenient for you. This is just step by step how we're doing. Just a P+, which is one of David's recipes, but pretty well the basic principle of this. You can add any mineral as long as it's in the right ratio. They're all pretty well the same. I've sort of played with cutting back the molasses and the milk and upped it and downed it, but we've sort of come to a happy medium. And it's we're probably half the rate of molasses of what David's got on his recipe, but it's still brewing just as quick, and it's pretty efficient in summer. Then your minerals. So sea salt, you want probably the most non-mucked with sea salt, so it hasn't been iodized or anything like that. Tech grade, so your MKP, which you just get from any rural store. He recommends Campbell's, which are awesome because they're going to actually dissolve. I've been having a fair bit of success through Redox products. They've got most things I want these days, so I can just tailor make these to what I need. Micronutrients, 5 kilos. So you can either, if you know you're deficient in a few things, you can probably divide that up into your 5 kilos. This is a very simple recipe. You can probably up these a little bit, but yeah, I'd recommend starting off with this and then working from it. Agitate and make sure all your minerals are dissolved. Otherwise, you're going to have all these little bits of granules sitting on the bottom of the tank, and that's where the goodness is. So you do really need to try and get them as dissolved as possible. And then when you are putting them out, I try and agitate it again, just so it really does stir it all up. So hopefully it goes out in the tank. Minerals, yes, most people probably realize the calcium and phosphorus are very incompatible. So you just got to be a bit careful that you don't add too much of those two things, especially at the same time, because they'll just lock up and then won't be available to anything. So yeah, I prefer to brew those two separately altogether now, and then add them when we're going to put them in the tank as they're going out. Boron can be another hard one with that. So adding the cab, so 100 litres of your cab, add some milk, more powdered milk, seaweed. You've got a lot of little biostimulants and stuff in there, yeast to speed it up, a calcium thiosulfate, super soluble, awesome. Yeah, it's really plant available. We've tried with a few bits and pieces, and we're always trying to try and find a better way of getting some liquid calcium, because calcium is deficient in most plants and it's not functioning. So the more of that sort of calcium we can put out, the better we are. And it is very expensive to buy in a liquid available form. So yeah, I think if you can find a good source of thiocal, yeah, you're doing pretty well. Magnesium sulfate, that's just Epsom salts, and yeah, they're pretty cheap. Yeah, mix everything together thoroughly, top up with water. Air gap is critical. I have blown up an IBC with not enough air. I got a bit greedy trying to stick a bit too much in there, and it just blocked up the airlock valve there. You can see in that picture that it's pretty crude, just the kefir bottle, a bit of hose and a barb at the top of the tank. And what I have noticed on a hot day, they really bubble hard, so they will often blow out a lot of the water. So you've got to every week at least go and check the water in the bottles that it hasn't all disappeared, otherwise it's not going to work. Mainly in summer or when it's really active. Yeah, seal for around 60 days, and yeah, you'll see, because they'll just slow down with the gassing, and then yeah, pH of around that 3.5 is what we're aiming for. So yeah, these are some trials I've been doing. This is what we've sort of come up with. I got this off a Vic No Till member. Yeah, some of the maximum stuff per 100 litres. When you're adding any of this sort of stuff, it's best to do it in small quantities, because if you just tip like a whole 25 kilo bag of sulfate in, you'll often kill the lacto. So you need to, I usually just put a couple of kilos in every day, and just, if you've got to keep feeding a bit of molasses and milk to make it, keep feeding it, that's the way to go, because otherwise, you're just going to end up with it all on the bottom, and it won't have attached to a carbon source. Application and rates. So we put these out through conventional boom sprays, so you've got to filter it fairly fine. We're now putting it through a 4 mil filter, and then it goes out in the taint fert, which has a 6 mil nozzle. That's a 4,000 litre unit. We've got a 500 litre unit, so when it's too wet and stuff, we can get on the paddock still without making an absolute mess. Super handy for your compost extracts and stuff as well, because we can put out that large particle, and the biology is actually landing on the leaf rather than being stuck in the bottom of the filter, and then you're just throwing it out. Yeah, so with our SAP testing, like we'll put out a spray like that, and then a day later we'll test. Then we'll have those results back, and then we know exactly what was like, if it actually got into the plant, or you know, what more it needs. And had really good success with that this year with the magnesium and boron. We've been super deficient, and now, yeah, they're actually back to optimum levels, which is exciting. The calcium is my next year's project, because it's still very hard to get into the leaf. Yeah, roughly 5 to 10 litres a hectare of bio-fert, depending on how many raw minerals you can actually get into that bio-fert, because you're sort of trying to work out like how many kilos a hectare you're putting out, or it's more like grams a hectare, like 300 to 500 grams of the actual raw mineral. And that, yeah, that's sort of the way we judge that. You can see the calcium, that's a gypsum calcium trial I did on a sunflower earlier in the year. You can see the big particle droplets on that plant there. Yeah, that's, so yeah, just a few acknowledgements. Thanks for David and Soilland Food. He always said to share the, if we can share the recipe, we've got to acknowledge him, so that's easy. And Vic No Till, we are very proud of farmers helping farmers, and that's the way we sort of work. You can call anyone up anytime and ask him a question, and most people will be able to help you out. Thanks to Courtney and the team at Soils for Life for putting this on. It's great to see so many people and lots of familiar names and faces from New Zealand and locally. It's very good to see. Thank you for attending, and happy brewing.
Thank you, Callum. That was awesome. Okay, question time. We have stuck to time. Well done, guys. Okay, so my first question is around fluoride in water. Does it affect a brew? I assume that's a yes. You would want non-fluoride water?
Look, if it really depends. The fluoride, anything in excess can impact. Usually we pay more attention to chloride or even sodium, things that can increase the electroconductivity. But yeah, that's a funny question. I've never thought of fluoride. Do you had any thoughts on that, Caleb?
I haven't experienced, like, we're nowhere near town water, so yeah, I haven't really had the fluoride or that side of it. But I think rainwater is always optimum if you can get it.
Yeah. What we normally suggest in treated water is always letting it, so even if you're doing an aerobic brewing or anaerobic brewing, is to let the water bubble before, especially if you're using town water. Even let it sit, let it bubble overnight, so it can actually gas off any of the gases that could actually be impacting your biology in the future. So that's what I would probably recommend. If it's treated water, town water, that's probably the best way to go.
Feel free to add a follow-up question back, if that doesn't answer your question. Rhiannon, how does high soil acidity and aluminium affect the biological activity of a biofert once applied?
You want to go first, Callum?
No, after you.
Okay. Look, acidity definitely. It's a common question actually for farmers asking, what should I do? Amend my soil and try to correct the base saturation, correct my pH, or should I go with the biology? I think if you got soils where you have really acidic situations or very alkaline, or you have high aluminum levels, I think you better sort of focus on your soil chemistry a little bit. But what you can do is, for example, you mentioned about really acidic soils. So as I mentioned, bacteria, they prefer more alkaline environments. But if you're doing bacteria and you want to put out like effective microorganisms, they're very resistant type of group of microbes, and they can survive in acidic conditions. So this is something that you could think about when you go into a very acidic soil. The fungi would thrive very well, but the more you keep pushing to a very acidic soil, less biodiversity you're gonna get, because you're gonna be restricting to only those ones they can handle that type of environment. And with the aluminium point, aluminium can sort of be toxic to biology in really high concentrations in the base saturation. But I think the biggest problem is in soils where the aluminium start going above, say, 7 or 8% on the base saturation, or even further than that, this really limits the root growth. And when you have root growth development, poor root growth development, that's the best environment for the biology. So if you're restricting your root development, your biology is not going to be in good conditions, because they use a lot of the carbohydrates and sugars being produced by the photosynthesis from the plants that are exuded to the soil, and that's where the biology lives, right around the root. So I think the aluminium could be a problem to the root system that could actually impact your biology as well. So probably focusing on amendments to try to adjust that aluminium and reduce that in the long term, it's crucial.
I'll sort of play devil's advocate a little bit to most agronomists with this one. I think if you focus on one thing like your aluminium or pH, you'll get too caught up in it and it'll end up costing you money. So small amounts often, so calcium, energy, that will stimulate that root growth through the aluminium layer. But yeah, I don't think it's one or the other. I think it's both. A lot of people get caught up on the acidity of your soil, but if you can really try and balance that plant out, that will buffer a lot of that out, especially with your calcium. And if it's got good energy to do that, I think you'll find that that's probably going to be a hell of a lot cheaper than putting 10 ton of lime out.
What kind of pH are you dealing with, Callum, or have you seen the pH shift at your property over time?
Here we're at about 5.4, but yeah, back where I grew up, we're in the sort of low 4s, and we put out a little bit of lime and with little avail, and then as soon as you started putting a bit of calcium and stuff out, it did slowly start coming up on its own. It's probably just as much about getting that grazing or whatever you're doing, putting a bit of compost down to create the humus on top of that soil to help buffer it as well.
Thank you. Rhiannon, feel free to jump in if you have anything else to add.
I know where Rhiannon is. She's literally,
Who have we got next? We have a comment that the recipe seemed to be a lot more than 1% of what Marco was kind of advocating for, but I think that was in reference to the culture.
I think Callum had a recipe where he had molasses for 1,000 liters, 100 liters of molasses, and then milk at 30. Because the milk would be more like an inoculant, not like a food source that you use, right, Callum? Or
correct. It's the protein. Yeah. So you're probably talking more about the actual mineral, which we're still over 1%, but you were also talking about aerobic, where we're talking about anaerobic.
Yeah, exactly. That's the big difference. The 1% is restricted to aerobic brewing, because once you start putting too much food, you create a really big spike on the population of microbes. And if the microbes, they don't follow the availability of nutrients, of oxygen, it gets to a point where the population is too big, not enough oxygen, they start collapsing and dying. So that's why you have to maintain to that 1% food source to avoid it. But in aerobic brewing, the story is different.
Cool, thank you. Bob has asked, where do you source the 1,000 litre fish scraps from, Callum?
We've been getting through Sampi, which it's sold as a fish hydrolysate, but it's not completely broken down, so that's how we get it to double. But yeah, there's heaps of people selling fish hydrolysate. The Sampi stuff I liked because it's actually got selenium in it.
And do you have any advice on how to source a kefir mother?
I can get that through to you and you can send it out on email. Yeah.
I feel like Facebook Marketplace is a good one for that. Jessica has not heard of calcium thiosulfate before. Do you know if it's a certified organic approved input, as per the usual rules where you can demonstrate a deficiency? Calcium nitrate is not allowed, so she does struggle to find foliar calcium sources.
I'm not aware. I don't think it is organic. It's a sulfate, so I'd imagine not.
I don't think either. It depends on the certification as well to check if it's a restricted or non-allowable input. But something that we probably can double check. And
yeah, I can confirm that for you, Jessica. From memory, I feel like liquid lime was kind of all there was for the certified organic folk. And there's recipes for turning lime into a more liquid form as well.
Yeah, calcium is definitely one of the hardest organic element. Calcium and phosphorus, because it's chemistry, it's really hard to get naturally extracted soluble calcium and phosphorus. So usually you end up really options is liquid lime or even the micronized lime.
Yeah. James from New Zealand has just started to experiment in trying to make calcium acetate. Oh, this is kind of what we're just referring to. He's making calcium acetate by using lime flour and acetic acid. He was inspired to try this after seeing a recipe using eggshells and vinegar. Okay for a garden, but impractical on a larger cropping farm. He's measured the calcium using a handheld calcium meter and achieved 3,000 parts per million. Would be interested in the panel's thoughts.
Funnily enough, I got one of those recipes sent to me about a month ago, so that's on my list of things to try. Pretty much exactly that.
Yeah, we should we'll try and find that recipe for you too, Jessica, because that would be up your alley. Callum, are you spraying the bio-ferts on your perennial pastures, and if so, when? From Donna.
We are doing it as soon as we can get on the paddock pretty well when it's green. So usually May is where we're aiming for. And then again, if we've got a second chance, we've been going since August again. Yeah, we put a last load out today.
Marco, I didn't ask for your input on the calcium acetate. Did you have anything to add to that? Sorry.
Well, the calcium, it's I think the calcium, a really important thing is trying to get the calcium at the right time. It depends what you're growing. I mean, in pasture, you probably in moments where you got more temperature and more biological activity to improve the availability of the calcium. In a broadacre scenario, it's usually important to try to put some calcium prior to the flowering stage. Usually a lot of crops, they get really slow on the calcium uptake once they head towards the flowering period of the crop. So trying to get that early, it's probably a good moment.
Hopefully that's helpful, James. Terry, after a few years of biofertilizer, are you measuring any changes in the soil tests? Are there any changes to the total nutrients? Great prezos, much appreciated.
We haven't tested again this year, and we've only been putting bioferts out for 3 years now, so I wouldn't expect to see a huge change. The amount of change is probably just going to be in the fluctuation of your testing anyway. So yeah, I wouldn't imagine it would have changed too much, but yeah, I'll let you know when whenever we do our next round of testing, which won't be till next year.
Have you done any Brix reading or any biological readings on what you do?
Yeah. So our Brix, like every one of our multi-species crops pretty well this year was 17 at least. In all the cereals, our brassicas were around the 10, and then we were getting them up to 15. And then they'd sort of drop back to like 13, and then we'd bang out another one, and we were up to 15 or 16 again. So yeah, consistent rise in the Brix levels.
And do you, so you mentioned you haven't done the total nutrients yet, or have you done in the past?
Yeah, we've done them already. I just haven't done a second one. No, I don't imagine they will have changed a huge amount with that little amount of nutrients going down there.
Oh yeah, not on the totals, but probably on the available nutrients. You might have a sheet on the availability of the nutrients. Because the totals is only with larger inputs, really. But on the availability, definitely a totally different scenario.
Yeah.
Could you please advise, Callum, what multi-species blends you're sowing? Is it bare seed, and do you do seed dressings?
Yeah, so all our multi-species were from down under covers this year. Grant Sims in Lockington. Yeah, all the seeds are bare. We had a very extreme seed dressing of Marco's Boss and Graham's State. I think it was probably a little bit too much. I haven't seen any advantage of it, so I think we'll probably cut a little bit back. We'll probably go back to just a Nutri-Soil and compost extract and maybe some fish or something. But yeah, we were putting about 12 things on the seed, and it was fine, but you know, I couldn't see any difference at all in the root growth or anything. We did sort of one paddock with and one paddock without, and yeah, I think simple is sometimes good, because as Marco said at the start, if it's too complicated, you won't do it. So I think, yeah, back to basics for that.
A little plug for our biological seed treatment practice guide as well, which is online. We've got a case study from Carol and Brian Fitzpatrick, who might be, I think I saw that they were online, maybe. And Will McInnes up in Queensland, so that might be helpful for you. Where are we up to? Oh, there's a comment with the Sampi fish. Oh no, it's a question. Could you explain, Callum, what you're doing with the 1,000 litres of Sampi fish, as they've never experienced it to not be broken down?
Yeah, well, like it's not broken down. It just can be broken down a lot further. So yeah, it does come with the odd little lump and bump in it. So yeah, when it's finished going through the process of adding the lacto, adding some molasses, I can send that recipe through to you, Courtney, and that can go out on the email, because it's really, really simple.
Cool, thank you. Bob has asked a question around SAPs. When should they be undertaken in the season, and how many times?
Depends what your budget is, I think. Yeah, we've aimed for 3 this year, and I think that was a really good guide for us. But yeah, I think depends on your operation and in your context. Like if you're growing a grain crop or something you're going to harvest, it's probably a lot more important to do 3, where we're just doing it for our knowledge and trying to improve the paddock. But yeah, it's not as crucial, I don't think so. Yeah, what do you think, Marco?
Oh, it's again, it's just monitoring the outcomes. It brings you confidence as well. if you're doing the right thing and when you're doing. But I would suggest doing the timing. I think it's probably maybe we can share later on guidelines when to do the SAP testing and timing, because if you correct it on the wrong time, the day, for example, the results can fluctuate quite a lot. So what you're doing with the SAP testing is really trying to be to give you the true story of what's happening in the plant at the moment. And I suggest doing at moments where you have some active growth in the plants, just to see when they're active. And if you're using that to monitor what's going on, just give after you do a biofertilizer application, give at least 1 week till you do the SAP testing.
Thank you. There was a comment from Katrina and Steve Sanders that the Sampi fish hydrolysate is derived from tuna, and they use a fish hydrolysate derived from multi-species ocean fish. That's interesting. And then Terry has a question. Have either of you got experience with Johnson Su composting?
Yeah, I've done 2. One failed, one worked. That was probably my fault. Yeah, I'd like to do some more. That's pretty well where I'm up to with that, because yeah, the results were good, but it's a long process.
Yeah, it takes up to 1 year to get to a good Johnson Su. Really good. There are really good responses with compost extract using Johnson Su. I think that's one of the best ways to use it. And it's really great for what I was discussing about the stubble digestion. It's totally fungi dominated, and it's a really good one for you to increase that fungi to bacteria ratio. Can be applied again the same way I mentioned, or as a liquid inject, can be full spray, can be sprayed as a stubble digestion application. It's probably the cheapest way for you to get some trichoderma domination for the stubble digestion, because trichoderma, if you buy commercially, can be quite expensive, of course. So if you harness this trichoderma in the Johnson Su and extract the biology to do as a stubble digestion application, can be quite cost effective too.
Cool, thank you. Any final questions? I have 2 questions. Marco or Callum, can you explain why we are aiming for a pH of 3.5? Like, what is the mechanism that makes 3.5 the ideal number for a biofert?
I think one very important component of that is when you're brewing microbes, you always open space for contamination. And E. coli, for example, and Salmonella, they're big ones. And the same way as, for example, other effective microorganisms, the Salmonella and E. coli, they're actually facultative. So they can survive in an aerobic environment and anaerobic, but they can't survive if you push your pH under the 3.5 mark or 3.7. So one thing that you're doing with that is already trying to get rid of contamination, because you're looking for your lactobacilluses or your beneficial microbes. So this is one of the point. And you got any other thing to add, Callum?
Probably the plant, yeah, uptakes it a bit better at that more acid level as well. That's probably, yeah, all I've really got on that one.
But you're measuring your biofert at the end of its brewing, or before, like at the end of its brewing cycle, to check that it is at 3.5?
Yeah, give or take a bit, yeah. Sort of work under, as long as it's sort of, yeah, around that 3.7 to 3.5, somewhere there is good, yeah. And
what would you do if it wasn't? Like, what does that indicate? What do you have to do then?
Probably put the lid back on. You might have to feed it again to try and stimulate the biology, and let it keep brewing.
I had situations where clients doing effective microorganisms, they actually 2 different situations that probably might be a good one to bring up here was temperature wasn't enough. So what we try to do is just to push that temperature up, and as soon as the temperature went up, within 2 days, it went from 4.5 to 3.5. So increasing the temperature is one, and the other had situation where a farmer was trying to brew effective microorganisms, and we had to do sort of an investigation type of thing, and we discovered that he was using a molasses that was it was actually watered down molasses, so it wasn't very runny. So when he added the molasses at the rate that was suggested, there was not enough food for the biology to really get to that dominancy in the brew. So what we went and did is just adding more molasses to the brew, and then the whole brew kicked in again. So there are a few things you just have to really identify why that pH is not going down all the way to 3.5, and then you target it, and you should be good to go then.
Thank you. Brian's made the comment that after brewing the fish, it makes it a more stable product, less likely to react with other things in the spray tank. And Bob has asked you, Brian, how do you brew the fish? That low pH of 3.5, is that for aerobic brews too, Marco?
Sorry?
The pH of 3.5, does that apply for aerobic?
Nah, that's for the effective microorganisms only, because as if you're doing an aerobic brewing targeting fungi, then you would be chasing something a little bit more. Bacteria would be tracing anything more alkaline above the 7 mark. So it depends. That's more for effective microorganisms. For lactobacilluses, as Callum has been doing, that's the 3.5. That's what you aim.
And Ruth has asked, maybe we've already covered this, are you monitoring the pH of the entire tank as you spray out, Callum?
Depends on what we're doing, not always. Definitely, if there's chemicals going out every time we do that. But yeah, that's as little as possible. So, but no, I haven't been. I probably should be. Yeah.
Cool. And then I have one last question. Callum, with your correct me if I'm wrong, you're spraying out your bio-fert and then the following day you're doing a sap test, is that right?
Yeah, a day or two later usually. Yeah.
Like, how do you know that it's not like residue of the biofertilizer on the leaf that could be tainting the sap test, or is it?
Well, from what I've read about the sap, if it was a tissue test, that would probably be tainting it. But being a sap test, it shouldn't be, because it's actually being squeezed, and it should have been really washed probably as well. So by rights, it should just be what's in the plant. We've waited a week and a few days. It just sort of depends on the timing of when you can get this spray out, really more with wind or weather, and then it's got to be earlier in the week before we send it usually, just so we know we're going to get it there, so it's not going to sit in the mail on the weekend.
One thing that I usually suggest is just giving a few days to do a leaf testing, because or sap testing, because some nutrients, they take different timing for being taken up by the plant. Even though they're chelated or complex with biologies, nitrogen, for example, urea, within half an hour, a big fraction of the nitrogen is already taken up, and other nutrients, they might take up to a few days. So if you do that gap, you're probably going to be, but then that's the thing, it's the tricky part, as Callum mentioned, it gives 1 day and 5 days. It depends on the timing you can do it. You have to do what you can do. Leaf analysis, even though if you're doing 1 day or 5 days, is already going to give you really good and beneficial information anyway.
Yeah, cool. Are you using sticking agents, Callum? Like, what do you call them? Stickers?
No, we have in the past. I've used some of the NTS products, but yeah, not all the time. It does depend. Like if there's more insect there and stuff, we will. But yeah, definitely not all the time.
Fantastic. Well, if there's no other questions, Oh, Brian's responded about brewing fish. He adds molasses and the cab culture, locks it up and lets the microbes do the thing.
Yeah, the same recipe pretty much.
Yeah. Perfect. Well, thank you again, Marco and Callum. That was awesome. We have heaps of resources to pass on to everyone. And yeah, please stay in touch, everyone. Let us know how your home brews are coming along. And we'll be following up with an email with a short survey as well. We'd love to have your feedback on how you found tonight and if you have any other topics that you would like us to dig into. Thanks, guys.